You have gold in your hand, you don't need to sell it.
Yeah.
It will be sold by itself.
Yeah.
So that is exactly the variety should speak for itself. Keep the farmer in the front.
Yes.
Okay, don't think that what you are doing is for just getting some publication and some products and just for the sake of release. I am not for.
So what keeps you grounded and makes you come back again and again on the field to do this? Like what drives you in a way?
The key, one should be passionate first. Any person doing any work for that matter, not only agricultural science, anything. First, that passion should be there. If you don't have passion to do something, you don't do that even.
Everybody want to go in the medical and engineering field, but after not selected in that, then we choose agriculture.
Yeah, but in my case, it was not.
Okay.
Hey guys, welcome to a knowledge-packed episode that delves into the future of sustainable food production. So today we privileged to host eminent scientists from IRRI. Join us as we explore cutting-edge research and groundbreaking insight in the field of rice science with our distinguished guest. He completed his bachelor's from Punjab Agriculture University and master's from IRI New Delhi and PhD and then moved Iran. So without further ado, let's directly jump into the podcast.
You don't cut by sickles, right? There are machines to cut it. Okay.
So today we have a senior scientist with us. I want to request you, please introduce yourself, sir.
Yeah, I am Jauhar Ali. I am the head of the Hybrid Rice Development Consortium and also the leader for the hybrid rice seed technology for seed industry unit. This is we call it as hybrid rice technologies for seed industry, HRDC unit, and I head the hybrid rice development consortium.
We are so excited to having you on this podcast, and sir. Whenever we saw some Indian scientists, we are catching vibes like home. So without taking too much time, let's get directly jump into the podcast. So our first question, starting from the beginning, just put some glimpses about your childhood, from where you started.
Okay. I was born and brought up in Delhi actually.
Okay.
And a place near Pusa Institute, you must be aware of that. So we were residing in Narayana Vihar, which is very close to Pusa Institute. And my childhood, if I remember, we used to walk into the Pusa fields and collect for some plant or some insects during that time as a childhood. And very familiar with that place. So that was the best destination as a childhood to go around. And so right from the beginning, I had a lot of interest in agriculture.
Okay.
So that is probably why you chose agriculture as a career?
Yeah. So if you, in generally, people opt for agriculture after they don't qualify for medical or engineering, then they will opt agriculture.
That's the case with most of us.
Everybody want to go in the medical and engineering field, but after not selected in that, then we choose agriculture.
Yeah, but in my case, it was not.
Okay.
So in my case, I joined after matriculation itself.
Okay.
So I didn't wait till 12th class and then get my exam results and then joining. There was a provision in Punjab Agriculture University to tap the talent early on.
Okay.
They knew this thing happens. So they introduced this course as 5-year course after matriculation.
Okay.
So, and that degree is called as honors, BS Agriculture honors. And that 1 year they will train you on a particular field like crop sciences or crop protection and like that. So they have some disciplines. So I got trained in honors in BS Agriculture honors in crop sciences.
Crop sciences.
So that's why my starting interest itself started on crop breeding and other things.
So you did your bachelor's from PNU, if I'm
not Punjab Agriculture University is one of the very renowned universities from where Dr. Khush, a lot of great breeders came out of that institute. And Dr. Brar, Darshan Singh Brar, many came from that institute, that university. And The PAU, when I joined, it was in 1983. My age was only, at that time, 15 years old.
Oh,
That's
When I
quite
joined my,
young.
yeah,
That's quite
so,
young.
yeah, so in that 5 years I completed, in less than the 5 years I completed in 4 years by advancing my courses.
Yeah.
So by 19 and a half or something, I finished my BS Agriculture.
Yeah.
And I joined a master's degree in IARI
Yeah.
for genetics, and then PhD for in 1993.
Yeah.
So the point why Punjab Agriculture University is so important is very important from the aspect that Dr. Kush had come when I was in just second year, I think,
Oh.
in BS. He was going to introduce IR36, I remember that, and he was going to talk about IR36 rice variety, because that was a very big achievement by Dr. Kush. So we were sitting in the first few rows of the auditorium and watching him, and it was such a great pleasure to see. At that time we didn't realize the greatness of Kush at that time, but still we are all students, so we were very excited to see him. So that really motivated me to not to leave crop breeding and genetics.
Yeah.
So I pursued very systematically, took those courses, strengthened myself.
before listening this conversation, we discuss on one question, like you are always student, but now I have no question on this. I just want to ask, in India you completed your masters and PhD from IRI,
Indian
and
Agriculture
after
Research
that
Institute.
you go Iran. So
Yeah.
Yeah. How was your time in Iran? Like you were head of breeding rice there?
Yeah.
Yeah.
Okay.
So-
What happened is I joined IRI as a postdoc in '94. After my PhD, I was hired by Dr. Virmani as a postdoctoral fellow for two-line hybrids.
Okay.
So you should be aware the background of two-line first.
Yeah.
So at that time, Japan discovered in 1992, and they reported in the International Rice Genetics Congress at that time about this discovery of TGMS in Japonica background. So immediately, this was 1990, I'm sorry. And then this problem came to IRI, and there were certain staff from ICR went to Japan to negotiate to get that gene. And the price of that gene was $50 million to transfer from Japan.
Quite similar.
So they did not succeed in getting it. Then the problem came to the IRA students, can anyone take this problem and do a mutation study? So I said, I raised my hand, okay, I will do it. And we started doing very seriously on the mutation breeding, because to find a particular gene is like searching a needle in the haystack, right?
Yeah.
We have 55,000 genes. How are you sure that you will hit the right gene?
Yeah.
Yeah.
So we did a very good background why we should pick a particular set of material and screening protocols. Eventually we succeeded in identifying 8 TGMS lines, and we reported it, including one reverse TGMS even.
okay.
So this was the first time in the world to produce in indica background. What Japanese did was in Japonica, but we did it in indica background, for which I was awarded the Jauhar Ali Award also at that time. And when we did this, so IRI was very attracted to this topic. So Dr. Virmani invited me to lead the two-line hybridized work in IRI.
Yeah.
So that's how I first came to IRI.
So you
And after that, during that period, the understanding was I had to leave for after the 1 year of my work. I left to TNE to join as an assistant professor in Tamil. So I was given the position without even attending an interview. So people have to go there, interviews. But my publication record was good. Every other score, I scored better without even going for interview. Interview was like 5-10 marks, I think. So still I was in the top 5 of them. So eventually they gave me the position. So I had to leave to join that position. So in 2000, when I came back again, I got the second postdoc in 2000. So 2000 to 2003, I was a project scientist at that time. and that was on molecular breeding. So we are the first batch of molecular breeders to work on SSR markers, and that's how it's. So in 2000 to 2003, when we achieved this, there was a position again coming under Dr. Virmani as International Research Fellow. I was a candidate for that as well, but unfortunately I couldn't get it. So I had to leave IRRI in 2003. So one of our agriculture ministers from Iran had arrived in IRRI at that time. So he was congratulating me as if I am going to join IRRI as a hybridized breeder. So he said, "Why don't you join us?" I said, "I'm already likely to get it here." Then he said, "Okay, if you don't get it, you come there and help us." And that was an invitation from the minister himself. So that minister is Behzad Gharyazi. He was a postdoc at IRRI prior to that. He did his PhD at that time, and later also his postdoc. But he was known to me when I was doing my postdoc at IRRI in '94. So it was coincidence. He became a minister later. So that's why he said, "Why didn't you come?" Because I used to teach him and train him also on breeding.
Seems like everyone who comes at IRRI does something good. In
So
life.
something it was there. So he said, "Why don't you come and help us to produce the same thing you are doing here? Now you have opportunity to do." And it was an open-ended check. Whatever money you need, do it. And as long you want to stay, stay. So it was very interesting. So I thought, okay, when I didn't get this position, so I left to, and this was under IRRI Iran program, still linked with IRRI, and 6 years I was based in Iran, in Rasht, a place called Rasht. Rasht is a very beautiful place in Caspian Sea. Now many of you may not know Iran. It's a temperate country. First of all, it's not a tropical kind of a desert, you may think, right? All of you may think it's a Middle East desert. So that's not the country like that. The northern portion is completely temperate, and snowfall happens there every year. And they have 0.55 million hectares, and all of them is irrigated. They produce the best rice varieties in the world. That's called Sadri rices. Its price in the market is higher than basmati. Its aroma is more stronger than basmati. But they are locally consumed, so they don't. No one knows about it. So I was trying to breed the Sadri hybrids for them, and also in Sadri basmati hybrid as well. But unfortunately, the key breeder recently passed away, called Drushti. So anyway, so he left, and I also came back after that. 6 years I was there, and then I was offered at IRRI again as the leading the Green Super Rice project. That's why I came back in 2009.
TGMS lines. Can you please explain a little bit about the importance of TGMS line in two-line breeding?
Okay. Now my PhD was in two-line breeding in 1990 to 93. So when I told you $50 million already story, right? So that is that much worth. But at that time, we had no clue, because we were all working on mutation studies in order to get that best TGMS line. So TGMS line is it's a recessively controlled gene, a recessive gene, nuclear gene, which is a major gene. And if it is carried by a plant, it will become completely sterile at high temperatures and become fertile under low temperatures. So high temperature means anything mean temperature of 26 and above, it will become sterile. So and anything below 24, it will become fertile. So this was the normal pattern. Now, using this principle, you can produce seed by putting the sterile plants under high temperature, the TGMS lines under high temperature. You can use that principle to produce the seed. And its own seed, it can be produced in cooler environments. So therefore, you don't need to run around and this much faster in production. And you will have 30, like almost one step is gone from 3-line to 2-line. 3-line you need A into B to make a A-line seed and A into R. Here is only one step. TGMS with any pollen parent becomes a hybrid. And its own seed is self-pollinated, so you don't need to, the cost of seed comes down by half. The heterosis goes by 10% above than the 3-line hybrids. So this is how the 2-line technology is good. So what changed from 1993 to 90 now currently after 30 years? What changed? Though 30 years, up to 30 years, nobody was pursuing this because all the time they were talking about instability, the temperature would go like this, temperature would rise, and you will not get a good production. So this challenge was again coming back to us. So in 2016 we did a serious comeback again. So I was at TERI, and at that time DDG Kim Doberman, who was our DDG research, he said, why don't you lead this area again, since you are the best in this topic. So I said, okay, on that, and we discovered A07, which is becomes completely sterile at 24 degrees centigrade. So this became the game changer. So so far 26 was the level, and that's why it was unstable maybe. But when you brought the temperature to 24, a line that will become completely sterile at 24, that means you can grow anywhere and it will be sterile. Mean temperature of 24 is very conducive for seed production. So therefore we produced this, and we created the TGMS lines, the pollen parents, the hybrids, all of them. We invited top 5 companies to the table. They said, okay, please come, and here are the materials. What you need to do is pay ERI $100,000 per year for membership to this elite class of group called 2-line study group. It's a sponsored research. This money will be put onto 2-line breeding, more breeding, and you will get a lead time of 3 years, or 4 years. So it's all, again, there's a guidelines for that, and very well arranged guidelines. So in 4 years, the technology would be then open to other people as well. But those lines which were as the starting point. So many companies, these 5 companies who came together, took the material in 2019, 2020, and today is 2023. They could not prove me wrong. So that was a challenge. That if you prove me wrong, I will return your money back. No issues. No questions asked. Yeah. If you find the technology no good, okay, we give back your. But 20% will be held and 80% will be given back. So, but till now we didn't get it.
Also, my next question would be, needs to make like 6 to 7, so that could be around 3 to 4 years. So that's quite a waiting time. So I want to ask about what was the first variety that you produced, and how was your experience with it?
Okay, the first variety that I released was Tirchi 2 in Tamil Nadu Agriculture University, where I was collaborating with other scientists, not myself. This was a rice variety, early duration. But that was a conventional pedigree breeding approach. Okay, so that takes quite a time, almost 10 years to release a variety. So I was just joined that time, and I joined from 95 to 2000. So during that time, my contribution to that program helped. So that was the first. But my own developed variety in ERI was the earliest was in 2015, I think, where I started releasing series of varieties. But in Iran also I released several hybrids and hybrids. So which one I should say the first?
Can you tell us about the first climate resilient variety?
The climate resilient variety was at ERI only. So we created a concept called green super rice breeding concept. So this green super rice breeding concept is a very important concept, and I would advise all the students, anyone listening to this, should dig deeper and understand the principles and the breeding technology behind it. It's all available in the internet. Go to ResearchGate or to Google it, do whatever you want to do, but go and see how it is done. So the green supervised concept is development of varieties that are multiple abiotic and biotic stress tolerant without compromising on grain yield and quality. And this breeding technology takes less than 4.5 years to get a fixed material. Generally, in conventional breeding, you make a cross between two individuals which are diverse, and you make a cross and you start pedigree approach. You need up to F9, F10 before you can even have stable things, and before it can be brought out for adaptive research trials and all those things. So that is the conventional breeding approach. But nowadays, even that conventional can be speed up by the speed breed facility, or you can have what you call rapid generation advancement cycles. But all that would not create the kind of varieties that we created already and released. So across the world, we released almost 71 varieties, and the last one was our hybrid 72, which was a two-line hybrid. So almost 72 varieties in less than 9 years, 9 or 10 years. I joined in 2009, so you can see. So that was that occupies almost 33 million hectares in a cumulative basis, like the area again and again harvested. And the most prominent ones are in India itself. So like PR 126, IIR 56 Dhahan, which is another variety recently released. We had the varieties that were released in Bangladesh were also released in India, like Biridan 75, Biridan 69, Biridan 99, Biridan 75. And in Binadan 17 GSR itself occupies 11% in Bangladesh. And this is also getting popular in India now. And much of the area is spreading in Assam, in the northeastern states. So India never had a very good Boro variety so far. And Boro area is almost 1.5 million hectares. And that area in the eastern sector is very good for these materials. Because of the multi-stress tolerance, they are spreading very fast. And a lot of work is going on at this point without doing anything, without touching them. Somebody's liking it, somebody's promoting it. So if you have gold in your hand, you don't need to sell it. It will be sold by itself. So that is exactly, the variety should speak for itself. If it's really good, it will pick its own momentum quickly. And this is how it happened in case of the green supervised released in. These are the inbred varieties. Okay, so I lead the hybridized program.
If I'm not wrong, I guess you completed here more than 13 years in IRRI,
Yeah.
So, sir, over the year, what are the transitions or what are the challenges you face in the rice breeding program here in IRRI?
Yeah. So when I joined IRRI in 2009, the biggest challenge was they were already very because I am aware of the IRRI's program right from 1990. I have almost seen 5 DGs in my different capacities. I came as a postdoc, later as project scientist. So I have seen many DGs and have worked with very great people like Dr. Kosh, Dr. Brar, Dr. Virmani, and many other people. I can't remember all of them, but there are so many great people out here in the first phase. So I know already the terrain very well. So from the day one, we know what to do exactly. So if I remember, when I was a postdoc in 2000 to 2003, we were already working on green supervised concept. So the introgression breeding strategy was developed during that time, but applied in 2009. So you should know that it's not that it came suddenly, that idea came GSR and GSR became a big thing. So its historical background is from 2000 to 2009, a lot of work has gone inside. Even between 2000 to 2003, there were almost 8 or 9 postdocs working in that lab. It was called the Genome Mapping Lab. I worked with Dr. Sikam Lee, who was the lead molecular breeder at ERI at that time. I had the opportunity to work under him. Strategy of breeding was not new actually. It didn't start suddenly. One day I feel like I have to do it this way. It's not like that. It went over several times. So we understood how many back crosses should we make, when we should select. So we did all these experiments before. Should we do BC1 or BC2, BC3, and then subject the F2s. So all these things boiled down to precision. In 2009, we knew exactly this is how to breed. This is the fastest way. And in 4.5 years, we'd make the design, and that's how we did it very quickly.
My next question is a bit more personal. So it's been like 13 years here at IRRI for you, and also a few years back, like 5 years around in Iran. So what keeps you grounded and makes you come back again and again on the field to do this? Like what drives you in a way?
Yeah, that's the key. The key, one should be passionate first. Any person doing any work for that matter, not only agricultural science, anything. First, that passion should be there. If you don't have passion to do something, you don't do that even. So I had the passion right from my childhood, I told you. So that is driving me. I don't bother whether you get recognized or you don't get recognized, whether you will be patted on your back. I don't mind, I don't care even. So that should drive. That is your first driver. And along the way, you keep doing the thing. Keep the farmer in the front.
Yes.
Okay. Don't think that what you are doing is for just getting some publication and some products, and just for the sake of release. I am not for the sake of release. We want a variety. Eventually, bring smiles to the farmers.
Yes.
Okay. Otherwise, the very point of being a scientist is defeated. You can have hundreds of varieties, but if you cannot put a smile on the farmer's face or increased income for his pocket, then we have failed miserably. So that is how you have to think. And if you are doing so, then that will motivate you to do better and better. And you will keep on doing better and better. So, for example, when I started with green supervised breeding, we already have almost 71, I told you, released, and then again we have 104 in the pipeline, still in the pipeline, and thousands in the boxes. Thousands. I'm not making exaggeration here, because there is no absorption capacity of the breeding systems. Like, for example, there will be nobody even accepting it to be nominated in the national system, because it's like flooding their thing. So they have to also the absorption capacity. The driving force is basically for any youngster to pursue should be the passion, first, number one, and hard work. So passion, if you have, automatically hard work, everything will follow through, right? Yeah. So just keep that. People should mind that they should be very passionate of what they are doing, and not doing for the sake of doing, but really for sake of the farmers in the farm. And of course, the food security and other things are going to be very serious in the coming days. And it all matters now that how we position ourselves to solve the global food problem. Sitting in IRRI, we think globally. We can't think country-wise. Okay, and we have to think collectively for the thing. If we don't do, who else would do?
Of course.
Yes.
There's no other organization. If IRRI doesn't do, you tell me which other organization can do it. So IRRI is in the best position, which is fair and trustworthy. Everybody will trust IRRI if it moves forward to execute something. They all support us, and we have no. So IRRI is in the best situation actually to lead this and execute it, because we have nothing to lose. We are not a non-profit organization. We do research to bring that value to the rice value chain in such a way that everyone in that stakeholders get benefited, whether it's a farmer, whether it's up to the consumer. So from the field to the plate, everyone in the line gets benefited in some way or the other.
Yes.
And since we don't have profit as our motive, whether including the hybrid rice, we don't. So any profit that even comes by some licenses, it goes back to research. So that way, one should not think that ERI is trying to get some funds, but that fund is meant for doing better science. Okay, so more science and more scientific products are coming out of it, and more intelligent questions to be asked and pursued, so that the world becomes a safer place by getting their food secure.
Yes, sir.
Okay, so this is very important actually.
Yeah. So in your lecture, you discuss a lot about climate change. And yeah, it's a question of worry. Everybody is talking about the climate change, and rice field itself contribute more than 19% methane and carbon carbon in the environment. So how we can reduce it?
Okay. Yeah. So everyone knows.
And I have one solution also. I don't know it's some solution. You are discussing in the class. Then I thought in my mind that if we are sown rice, dry seeded rice, then there is no flooding condition in the field. So there is no methanogens, bacteria that produce methane. So there is no methane production, I guess.
Okay.
So is this right?
Yeah, I'll explain that.
Flooding
to you.
Yeah,
Sure.
you explain. Yeah.
So first of all, you should understand that the methane is not only emitted from
Yeah,
rice.
it's not from the rice.
Stubble.
Rice is one component,
Yeah.
but the other components are livestock,
Yeah.
permafrost areas, which are very large actually. Other areas are much larger, so don't blame rice for that.
yeah,
Number
yeah.
one.
Yeah.
Okay, rice is a food crop,
Yeah.
and you can't do away with a food crop.
Yes.
What we can do is reduce the methane emissions. We can reduce the greenhouse gas emissions.
Yeah.
So greenhouse gases are what? Your
Yes.
CO2,
Methane,
your
and
methane, your
nitrous
nitrous oxide. These are the gases.
Yes.
So what they do actually, they heat up the
atmosphere.
environment. So when you heat up the environment, what happens? The glaciers, the permafrost areas gets melted away. More the permafrost areas melt away, more methane would produce.
Yes.
So it will be like an accelerator. You are stepping on the accelerator. So if we don't reduce the carbon footprint at this point, you are simply stepping fully on the accelerator, and that would destroy the global temperature balance. And if that happens, everyone will suffer, including human beings, animals, everyone, right? And that's not very far away. Whether we like it or not, it is happening. What we can do is we can reduce the speed. So how do we do our part from a rice perspective? Is how we can reduce to our whatever we can contribute. So rice is typically an aquatic plant, semi-aquatic plant, right? It loves water.
Yeah.
Now, how to do away with water is the question.
Yes.
So that means we have to breed something for direct-seeded rice. Now, direct-seeded rice is just like growing wheat or barley without water. And without water, how it will be happy? There will be a number of problems coming there, right?
Yes.
So it has a whole set of problems. If you grow dry, there will be nematodes there. There will be other insects, pests, weeds. Everything will come. Now, how do we have solutions for them? So it's a holistic exercise, combining breeding and agronomy to make sure that you are able to produce the right direct-seeded varieties, right? So direct-seeded rice varieties has to be bred,
Yes.
not by doing some
of
screening
course.
of already existing varieties and then I put it in direct-seeded and then I put it in transplanted, which is better yielding, and you make a decision. It's not like that. You have to breed, systematically breed under dry conditions and select and advance them every time. And there's a whole new set of breeding strategy evolved for the early segment. We put it in the early segment. And the early segment, the main focus is on dry direct seeding. Now, direct seeding can be two types, dry and wet. Okay, so the dry seeding is more useful for reducing the methane drastically.
Yes.
Puddled is relatively, it's a better option where you don't stress the plant so much, but you get the benefit of both the worlds, something like that. Okay, so So one should think, weigh the options, which is better and which is not, and what is most suited breeding strategy to produce the inbreds or hybrids around that. So currently this is happening. Our early duration breeder, who is PR126 currently, which is a direct seeded also, and also transplanted. It's a dual purpose variety already. It's occupying 40% of the area in Punjab, Haryana, of the geographical area, and its yielding is very high. And likewise, we released another IIR 56 Dhan, which is just 116 days total duration, and PR126 is 123 days, and PUSA 44 is 155 days, and it reduces time by almost a month. So 1 month of water is conserved, 1 month of inputs is reduced, 1 month of labor, everything is reduced. So this variety is making a big breakthrough in this terrain. Not only in India, also it is spreading to Nepal. It has been released in Nepal, and it's spreading very rapidly now. But now the question here is, we are using the genomic selection tools in order to reach that point. So, as I told you in my lecture also, the elite by elite crosses of early duration with all these traits for dry direct seeding. So what all it should have: early establishment, early vigor, coming out of the deeper depths, the germination should take place. When you throw the seed, it will fall deeper, and it should come out very easily from there. Establishment, uniform establishment, compete against the weeds. Anaerobic germination tolerance, that means when you flood it, it should not die. The weeds should die. Then it should have less nematodes, resistant to nematode resistant. So there's a series of traits. If all these traits are there, then only it will be a good successful inbred variety. And if you get that successful inbred variety, in hybrid rice, we'll use it as a parent straight away. So automatically the new hybrid would be also direct seeded hybrid. So we are also working on the sidelines at this time, also to bring herbicide tolerance as well. So herbicide tolerance is a very important trait, because we need herbicide tolerant genes in the rice plant. Whenever we spray certain herbicides, the rice plant will be resistant, whereas the weeds will be dead at post-emergence or even at pre-emergence herbicides. So that helps especially for post-emergence herbicides to control the weeds. So we already discovered certain mutations for that. Also we are working with other companies like BSF for Clearfield and Provicia genes, and we recently discovered the HIS1 gene. So all our hybrids are HIS1 gene resistant. That means it can resist, or you can say herbicide tolerant hybrids by default, because it is a native gene. It is present in our CMS lines and TGMS lines. So automatically all the hybrids that we bred out of them, those parents, are already possessing these genes. So only thing we are now verifying and validating now at this point, and soon it will be understood how good it is.
Yeah, just what would be your message to the students seeing this through the video?
Okay.
Yes.
Okay, that's very important.
Especially who want to make career in breeding.
Breeding as such.
Yeah, so breeding is something, first of all, those who want to take agriculture as their career, but make sure that you are prepared for it.
Yes.
Mentally and physically, and this is the most toughest of all the courses, right? Computer guys will sit under a lab with an air-conditioned lab and everything will be fine. Agriculture is not like that.
Yes.
You have to toil in the field, you have to go to the most of the time under the sun or rain or anything, but you have to be prepared. So be prepared for taking this career first. If you are ready with that career, then jump into it. Otherwise, stay away from it and go to do something else. Okay? Don't come half-heartedly.
Yes.
Okay? So take this career very seriously, because it rests on your shoulders. We have done our part at this point.
Yes.
Maybe another 10 years we'll do more. But beyond that, you are the guys who are listening to this. So we want that young students like you to choose career in breeding, crop breeding, which is the most important of all the disciplines. I can convince you in just 3 points for that. The first thing which I would suggest you is think that can you increase the existing variety that is prevailing in your near your farmer's field is say 5 tons, and you want to make it 5.5 tons, even half a ton. Don't you see that half a ton would feed how many people? So say even you create half a, and if you create a hybrid, automatically it will be more than a ton. So 1000 kg and 1 kg can feed how much people? 5 people, very comfortably, right? For a day. Then you can imagine 1000 kilograms would feed how many? 5,000 people, just on 1 hectare basis on a particular day, right? Now imagine you multiply in millions of hectares that the variety that you are cultivating, how much benefit economically you are bringing to the farmers and to people whom you feed indirectly. Your joy, your passion, everything will get rewarded by looking at that number itself. That, okay, I created this variety, this would feed this many people without even giving any money from your pocket, right? But the people who are consuming your variety, definitely they will bless you, right? And that is your reward actually. So that is exactly the point. So one has to think that way in order to get thing. And the second thing which I would suggest to the student is the breeding such varieties not only helps in understanding the molecular and the biological reasons why such a thing behaves. You have the opportunity to release a variety, understand the pathway, try to exploit that pathway for the benefit of the getting higher yields. When you get higher yields, it's not just feeding the people, it's also increasing the income of the farmer. So that increased income to the farmer would bring smiles on his face, and that smile means a lot to them. You don't believe, you just when you go to those fields, those who are proudly growing them, you listen to them, then you will see how happy they are. And they say, oh, this has happened this time, this happened, and they are so excited to tell you when they see the benefits of that. And if you don't do your job good, the same thing would happen also. They will put it in the trash can. Okay, so the varieties don't stand a chance. Even it may be released, but if it is no good, it will end in a trash can. Nobody will even cultivate it. So one has to think very deeply whom you are serving and serve them the best. Don't do half-heartedly. So put your 100% on that and see the best tools. Nowadays, the best tools are available. That's the third point. So we are living in an age of what you call genomics era, and we should say post-genomics era of rice especially. Today we can sequence any line with a very limited amount of money, right? You don't need millions of dollars. Now thousands of dollars is enough to sequence anything. And once you know the sequence of your materials, you can play with it, literally play with it. If you have that knowledge of how to manipulate it, that itself is so fun. And you know which genes are located where, how you can recombine them, and why we should recombine them. All that things makes such a important area that no other discipline can give you that joy. Because you are creating that unique product by understanding the genetic basis of that, and we can easily manipulate it in the interest of the farmers' high yield and income. That would drive, and then of course now ERI has already 3,000 genomes sequenced. We have another 10,000 genomes getting sequenced, and you imagine a time would come almost everything in the rice gene bank would be even sequenced. I don't see that even a challenge. Maybe we have 130,000 plus accessions, so who knows, that much money would come one day and all of them would, or the technology would become so simpler. The whole gene bank can be sequenced and anyone can use freely. So these are open resources. The good thing in science is nothing is held secret. The genome sequence is open. You can download any of the sequence freely. study them carefully, do allele mining, understand the genetics of them, use any population, think innovatively, apply the genomic selection tools, breed better and better. So, but that genetic gain one has to keep in mind. That means every 5 years or 10 years you need to jump your yield by a certain percentage, right? Otherwise, see, climate is changing. We can't just stay in the same place, right? And it will become worse, in fact, in the coming days. So you guys have to think that these 3 major points which I said, just remember them very carefully, and that should drive you to choose plant breeding or genetics as your career. It's not only for rice, any other crop even. The same thing applies. So if you like rice, go for rice. If you don't like rice, do for other crops.
Yeah.
With that, sir, I would like to say this was a very insightful session. Sorry for disturbing you this early in the morning, and it's been a great pleasure interacting with you. Thank you so much.
It's my pleasure.
Thank you so much.
And the key message again for you guys who are from this university. So what you need to do is keep that passion.
Yes.
Okay, what you are doing today is a passion.
Yeah.
It tells about that passion.
Yeah.
Don't lose that passion.
Yes.
Try to keep that going through. Okay, people just stop somewhere. Okay, we have done our job. Okay, now let's move on. That's all. You need to keep that passion, and that passion, how you need to cultivate it.
Yes.
Just like you cultivate rice, you need to cultivate this character.
Yes.
No, you will have a lot of hardships when you are passionate. When you are passionate, it doesn't come on your plate everything.
Yes.
You have to struggle. You have to do hard work. You have to be dedicated for it. You have to be honest. You have to report correctly. You have to change your character completely. Whatever you are doing now is okay, fine, but you may not be or may not be. But if you are going forward and choosing a career like that, then remember that one passion can change everything. Your character, your ability to think big, and always see the bigger picture. Don't see narrow picture. Nowadays, the world is showing you the narrow picture. Everyone talking small things, small things, small differences, small things. Think big. Think bigger than, think bigger. Don't think narrow. Okay? The world is not a big place. Okay? The whole world is a very small place. If you step out of the solar system, then Earth will be a dot. Right?
Yeah.
And if you step out of the Milky Way, it will be a dust particle. And why all the fuss about living on this planet? Right? So that's it.
Thank you so much, sir.
Thank you so
This
much,
was a
sir.
great conversation of us, and I'm sure we are taking something out of this with us back home,
Sure.
and we'll be back here someday with you.
You are welcome. Always welcome. Keep that mindset. I'm sure you will all succeed.
Yeah.
Wish you all the best.
Thank you so much.
Thank you so much.